So I call it intact cell approaches. I will touch on that. We need structural methods, meaning we need to be able to see what cells and their components look like. So there's a couple of methods that we will touch on. We need to think about the genetics, the instructions that drive cellular processes. So we need to look at how proteins are formed and what gene structures need in order to form those proteins. That's molecular genetics. And we need to think a lot about where and when we need our proteins to be formed and how that's regulated. That's the study of gene expression. We'll touch on that. And then finally, we need to understand the smaller components of the cell, so the proteins, the organelles, the membranes. So that means we have to take the system apart, isolate organelles, isolate macromolecules, proteins, nucleic acids, put things together again and see if we can get it to work. And if you do that, then you're a biochemist. What am I? I can ask myself. I'm most a biochemist, molecular biologist. I'm on this side. I also really like the microscopy. So we have to think about these different approaches, and everything really comes together for our integral understanding of cell biology. Multidisciplinary approach, okay? So let's first look at the intact cell approaches. Yes. Okay. So cell culture is one way in which we can study our cells. Hey, it's cell biology, so we work with cells. Okay, so why do you want to culture cells? Well, you want to look at individual cells, and cells in the middle of a multicellular body you cannot look at. Okay. So you can observe individual cells. You can access those cells. You can add chemicals and see how they respond. You can see how individual cells respond to signals. You can study, for instance, if you need hormones which function as growth factors for the cells to divide. Something you can study. And then the important aspect is that if we actually culture cells in a dish or in a test tube, we get copies of cells with the same genetic material. So we can split our cultures. We have one clone, we split it in two separate cultures, and we can do the treatment and control. We can say add the growth factor to one set and not to the other set. So we have the perfect control. The groups are identical. If you want to do an experiment like that with individuals, let's say you're working with dogs in some kind of a way, maybe you're looking at behavior, right? Well, every dog is different. So you have to have a lot of repeats if you work with intact organisms, because they're just so different from each other. You cannot make a clone. Right? So it's just a big advantage experimentally from the point of view of access and the point of view of reproducibility when you work with cell cultures. Now, some cells or some organisms are single-celled, microorganisms. Yeast is an example, many bacteria. So, what about those? We can grow those on media that we can control. Now, an important thing to realize is that if you look at all the diversity of life in the microbial world, it's estimated that less than 0.1% actually can be cultured. And even much and much less has even been successfully cultured. Most of the diversity of life on the planet cannot even be cultured in the lab. We can only see those organisms out there in nature. And therefore we know very little about them. It's not an extremely important little fact for this class, but if you're a biologist, you should think about that. The diversity of life is much bigger than you think. How do we know that those 99.9% of the other organisms are there? Well, we can find their DNA. Right? You can see their DNA, you can sequence that, we know they're there. We cannot figure out what we need to give to those guys in order for them to grow in the lab so we can study them. So keep that in mind. Even for the organisms that we can grow, we can often only grow them under a limited set of conditions, which might not be the most biologically relevant condition for those organisms. Okay, but still we can culture many, and from those we can learn. Typically we grow them on media that have a solidifying agent in there called agar or agar-agar. Agar is actually a polymer that is isolated from some algae that live in the sea, in the ocean somewhere. Actually, agar is an Indonesian word. And in the Indonesian language, the multiple, they just repeat the word, so agar-agar. I don't know why it's a multiplication. Okay? But so it's a polymer, and what's nice about it is that most microorganisms cannot degrade this polymer, so therefore you can make a solid substrate. Now, in the ideal world, you'll isolate your microorganism so you have a single cell somewhere on the agar plate surface, which grows up to a single colony. And that single colony technique is the most important technique in microbiology. Because you get clones of one cell that are identical. You can then put that in a liquid culture, you grow up many cells that you can study. And you can define the media, meaning you can put in whatever you want. You can put in only amino acids or only sugars or certain salts, whatever you want to put in there, or whatever you want to leave out, you can control it. And so you can study the growth requirements of your microorganism. So, what are the growth requirements for typical microorganisms and therefore actually for the majority of cells? Well, we need inorganic salts, potassium, magnesium, calcium, a couple of others. We need a carbon source. That means we always need a form of reduced organic carbon, sugars or fats, fatty acids, for the organism to break down in order to gain energy. There's only one exception, and that is the organisms that can truly photosynthesize. Because those you just have to give light and CO2. So we need a carbon source or light and CO2. Okay? So if you are an organism that eats a carbon source, meaning organic molecules, sugars, fats, you're a heterotroph. So guess what you all are? You are heterotrophs. And I am too. Proud heterotroph. So the photosynthesizers, the plants, cyanobacteria, and some green algae are autotrophs. They can make their own carbon source. But in addition, next to the salts and the carbon source, some organisms just need something extra. Maybe a vitamin or a single amino acid. I call it a goodie. So how do you find out what your organisms really need? Well, we can do experiments using different colonies on agar plates. And experiments like this have been very useful to start the process of biochemistry looking from a genetic standpoint. So we can ask the question, for instance, what does it take for a cell to make the amino acid arginine? So what can we do? Let's say we have a microorganism and it grows on an agar plate and forms these colonies. So these greenish, is it greenish? Greenish dots here are colonies derived from single cells. And we have here a rich medium, meaning something that's derived from the soup. Maybe you boil some meat and some yeast extract that has everything life wants, it's in there. So it has all the amino acids, all the nucleotide subunits, all the vitamins, everything. Salts are in there as well. So everybody grows, everybody's happy. Let's say we have plated here cells, plated means spread out, cells that have been mutagenized as a population, maybe by UV light. And let's say that in one cell the UV destroyed one or two genes that encode for a protein required for the synthesis of arginine. How can we find that one? Well, if we come with here this stamp covered with a velvet cushion, we make a replica, and that replica we stamp onto another plate. One plate here is composed of a defined medium of all the amino acids, etc., except that we leave out arginine, and here we have arginine added. So we get here, we hope, all the colonies that we have on the original plate, because we have all the amino acids and all the other food sources, goodies, nutrients. But here we have one colony that cannot grow. We see here but not there, where there's arginine missing. So we infer that that strain of microorganisms has a problem in its gene encoding a protein required to synthesize arginine. And so we can repeat this experiment, figure out how many genes are required for the synthesis of arginine, and then from there find out what enzymes are encoded by these genes and figure out the process. Okay, so a colony like this is called an auxotrophic mutant. In the labs this week, you will be using some yeast strains which are auxotrophic for a couple of mutants, and you will actually use that property to find out the mating types of yeast by complementing. You see how that works. So you will work with auxotrophic mutants. Auxotrophic means it needs some help in its feeding process. Troph means fed, to feed. Okay, so that's microbes. What about multicellular organisms? Our cells like to be in our body. They don't like to be in a petri dish. So how do you get them there? You have to separate the tissue. If we have animal tissues, we have to separate the cells. We can do that mechanically by shaking, but that often doesn't work very well. Or we can use enzymes that attack the extracellular matrix, as proteins that connect the different cells in the body. A commonly used enzyme is trypsin, a digestive enzyme normally. So we can separate the cells in a tissue, and then if we're lucky, we can incubate those separate cells in a growth medium, in a tissue dish, or in a culturing tube or culturing flask. And if we hit it right, if we have the right growth medium, the right salts, the right nutrients, the right growth factors, we may see them grow. Now, typically, for animal cells, the growth media are pretty complex. And what that means is we do not know the full composition. Even though 90% of the medium, as we prep it, is defined, meaning separate salts, etc., we can just all add, we often need to add a little bit of serum. Serum is the fluid in which the blood sits. And what is in the serum? Well, many, many things. So the growth medium often is complex if we think about animal cells. A limitation for animal cells is that due to an inherent limitation on the amounts of divisions that cells can undergo in the body, we can often not culture cells indefinitely. So we're talking about mammalian cells. The exception are tumor-derived cells, which have overcome this limitation, which are said to be therefore transformed. All right. So in animal cells, typically there's no regeneration possible. If we go from the cell culture, can we get back to an intact body? Typically not. This is because cells in tissues taken from the developed body are too far in their differentiation process. You cannot differentiate back to the types of cells that have all the fates possible. We have about 200 different cell types in our bodies. So from an embryo, we need to form 200 different cell types. And as a cell type is further and further specified, we lose the ability to form other cell types in that differentiated cell type. Okay? The second limitation is limited generations in culture, because of what I just mentioned. A lot of this is related to the replication of the ends of chromosomes. The ends of chromosomes are called telomeres. Telomeres means ends. And those telomeres require a special enzyme, telomerase, in order to maintain them. And that enzyme telomerase is not active in most cells of the body, which limits the generations that we can get in culture. Most cells want to form a layer, single layer on the bottom of the tube. It's not always true. Special cases are the cancer-derived cell lines. They're said to be transformed. There are two meanings of the word transformed in cell biology. One meaning, this meaning, means the cell is genetically changed so that it has become cancerous. It divides without control. The other meaning for the word transformed is if we introduced foreign DNA as an experimenter. That's not what we're meaning here. Famous example of a transformed cell line is the HeLa cell line, derived from, I think it was a cervical cancer, and the patient was Henrietta Lacks. There's a very interesting book that was written about this by a former CSU president. Okay, so cancer cell lines can grow forever, grow in suspension. The HeLa cell line, I think, is from the 50s, and it's a well-growing line. Plants are different. They can undergo many generations, and they can be regenerated. Okay, that's enough for you today. Stay warm, and I'll see you on Wednesday.